Hello, dear students. Myself, Sunita. Welcome to this session. In today's session, we shall be learning a new chapter which belongs to the unit 3, that is cell. So, in this chapter, cell cycle and cell division, we shall be looking into what exactly is cell cycle and how cell cycle plays a very important role in each and every individual, starting from the unicellular organisms until multicellular organisms. That will be the first part of the chapter. In the later part of the chapter, you shall be understanding about what exactly is cell division. So we know that cell division plays a very important role in our life for growth, for repair, for reproduction, for continuing the life pattern from generation to generation. Henceforth, each and every cell has to undergo cell division in order to perform various functions. So you'll be learning about the types of cell division, which includes the reductional division and the equational division, that is meiosis and mitosis, with respect to their different stages. And also you'll be learning why meiosis has to occur and what is the significance of mitosis. Okay, so we shall look into the first part of the topic, that is cell cycle, in today's session. So we'll understand first what exactly is cell cycle. Before that, we'll understand how the cell cycle came into existence, like who are the scientists who contributed for understanding of cell cycle and gave an idea what exactly happens during the cell cycle in each and every cell of an organism. So we'll understand what is the definition of cell division first. So we know that the process of formation of daughter cells from the parental cell is known as cell division, or in other words, it is also called as cell multiplication or cell reproduction. So you can even address it as cell division, cell multiplication, or cell reproduction. So these are the various names which have been given, but the meaning is same. So the process of formation of daughter cells from the parental cell is known as cell division, cell multiplication, or cell reproduction. Two important scientists who remarkably studied the process of cell division was Boveri and Fleming in the year 1879. So what did they study? They studied the process of formation of two identical cells from the pre-existing cell. Here, pre-existing cell is nothing but the parent cell, and the two identical cells were nothing but the daughter cells. So it is nothing but the process from parent to daughter cell. So the progeny is formed from one generation to generation, and this goes on, right? So there are two types of cell division in eukaryotic organisms, basically, which we'll be learning, that is mitosis and meiosis. So we'll understand more about this mitosis and meiosis in the next few sessions, where in detail you'll be learning what exactly is mitosis and what exactly is meiosis. But you can see in this diagram that this is the parental cell. So when the parental cell divides by the cell division, it is going to form the daughter progeny. The number of chromosomes, say for example, the number of chromosome in humans, it is 46. So the chromosomal number in mitosis remains same. It will be 46 and 46. Whereas in case of meiosis, which is called as reductional division, mitosis is called as equational division because equal number of chromosomes are going to be shared among the daughter progeny. Whereas in case of meiosis, when we look into the picture, you can see that meiosis will undergo twice. Okay, we'll be learning the steps later. But when we see the chromosomal number, the number of chromosomes will divide between the daughter cells. Okay, so henceforth, the reductional division will be occurring here. But if you see, 23 chromosomes have to again redivide. If it redivides, it cannot be 12.5. The number of chromosomes retain the same after every generation once it has undergone the meiotic division. So we'll understand why here it is 46 and why here it is 23 and why here 23 is remaining. And we'll also understand why 46 here and why 46 is in the daughter progeny. Okay, so now before understanding about the cell division, we'll understand what exactly is cell cycle, which involves cell division. So coming to the cell cycle, we all know that cell is the basic unit of life. So if a cell has to undergo the growth or reproduction process, then it has to follow a sequential pattern. And this sequential pattern, which is followed by every cell in our body, in all living organism, is known as cell cycle. So cell cycle is nothing but synthesis of the constituents of the cell, which eventually divides into the daughter cells. So that is termed as cell cycle. So if you see the definition, cell divides into two daughter cells and repeats the growth process. So this cycle of growth and division is called as cell cycle. So we can say it as cell cycle is equal to cell cycle equals cell growth plus cell division. Cell growth plus cell division. So the cell cycle was first studied by two important scientists. One is Howard and another one is S.R. Pillay. Howard and Pillay studied the cell cycle and proposed there are 4 stages in the plant which is called as Vicia faba, that is nothing but broad beans. So they first studied the pattern of cell cycle, that is the sequential events which take place during the process of growth and division in broad beans. Okay, it's a vegetable. So which belongs to the family called as pulses. And during the process, they identified 4 stages. So we'll be looking into what are those 4 stages, very important stages. So the 4 stages which was remarkable was, one is called as the G1 phase. G1 phase. In other words, G1 is called as growth 1 phase, or it is also called as gap 1 phase. So there are even other names for it. It is also called as first growth phase post mitotic division, or it is also called as pre-synthetic phase. So there are different names. We'll be learning that as we go ahead with each and every step. So this is the first step. This is the first phase, that is G1 phase. So G1 phase, after G1 phase, then comes the next phase, which is called as S phase. The S phase is called as synthesis phase, or it is also called as replication phase or DNA replication phase. And then comes the next phase, which is called as G2 phase. You have to write 2 subscript. G2 phase. So that is after S phase. So henceforth, G2 phase is also called as gap 2 phase, or it is also called as growth 2 phase, or it is also called as intense cellular synthesis phase. Then comes the next phase. The next phase is called as M phase. Okay, so this is the M phase. M phase is divided further into 5 important phases like prophase, metaphase, anaphase, telophase, and cytokinesis. I'll just represent it with the alphabets: prophase, metaphase, anaphase, telophase, and one more phase which is called as cytokinesis. So this represents M phase. All right. So the phases here are broadly classified into 2. G1 phase, S phase, and G2 phase altogether is called as interphase. Is called as interphase. The next phase, that is M phase, can be mitotic stage. Okay, it can be mitosis or it can be meiosis stage. So I hope this is clear to you. So cell cycle is nothing but, now if you have to explain the cell cycle, it is the sequence of events. So what are the events which take place in G1 phase? What are the sequence which takes place in S phase? What are the events which take place in G2 phase? And what are the events which take place in M phase? All together in a sequential manner is in a cell from time of its formation up to its own division into the daughter cells. So by the time it is going to form a cell and later it has to undergo the type of division. So this whole process is called as cell cycle, right? Now, the cell cycle, the time duration for the cell cycle goes on varying from one organism to another organism. Now we shall look into what are the duration which take place with respect to each and every organism, and how much time do our cell, the human bodies, how much time do they take to complete one single cell cycle. So we look into the generation time now. The generation time is the period required to complete one cell cycle, that is from beginning of one cell division to the next cell division. So just now we saw starting from we can say that from one M phase to the next M phase. So the phase which is present between is called as interphase. Okay, so interphase. So the time duration from one phase to another phase, and which includes the interphase, so that is called as the generation time, right? It is called as generation time. Now we'll see into the generation time as per organisms. So bacteria takes about 30 minutes to divide. So if this is a bacterial cell, it undergoes fission. So it divides into two, and the time duration for this will be 30 minutes. Okay. Likewise, in case of yeast, we know that yeast is also undergoing cell division. So here, the budding is a sexual reproduction, and this take place somewhere about 90 minutes. 90 minutes. And the mucosa cells of intestine in our body take place about 8 to 10 hours for the completion of cell cycle, that is for the division. Then human cell on the whole takes place about 24 hours. Okay, so the duration of human cell is about 24 hours, which completes a life cycle. So generally, in case of the human cell, when we have to grow a stem cell or in any other artificial medium, so it takes place about 24 hours to complete the cell cycle. So obviously the cell cycle will vary from one organism to another organism. So because we have seen here yeast, it is different, bacteria it is different, so it goes on differing from one organism to another organism. But all these events, G1 phase, S phase, G2 phase and M phase, it's universal. There is no change in the phases, but the duration of time is the one which we have to focus on. Okay, that is the only change which we observe. Now, coming to the phases in detail, we'll go ahead with the details of the phases. So basically, it's divided into two important phases. As I told you earlier, the phases of cell cycle is divided into two phases. One is the interphase. The second one is the mitotic phase. Interphase is the phase which is a longest phase. Okay, interphase is the longest phase. It takes about a lot of time, whereas in case of M phase or mitotic phase, it is short dividing state. Okay, so longest phase of the whole cell cycle means it is the interphase, and the shortest phase in the whole cell cycle. So when we talk about cell cycle, we know that it is divided into two phases. One is the interphase, another one is M phase. So this is the longest phase, and this is the shortest phase. Why I'm stressing and telling you is because again in interphase there are longest phases. Okay, among the three there is longest phase. So we'll see which is the longest and which is the shortest period in case of interphase. So first one is inter means between, phases means stages. So what are the events which majorly take place in interphase is what we have to focus on. So in case of interphase, it is the period between two successive mitotic division. As I told you, M phase, then comes I phase, and then again comes mitotic phase. So this is a complete cycle, sequential events which take place from one cell to another, one cell division to another cell division. So the period of active synthesis and intense growth takes place in M phase. See, interphase. So interphase is the phase which includes G1 phase, S phase, and G2 phase. So when we are talking about active synthesis, it includes all. So here the synthesis of the organelles may be, the synthesis of cytoplasm may be. So active division goes on taking place in the interphase. So in case of human cell, 23 hours is spent for interphase and rest 99 plus odd duration is spent for the next phase, that is in another 1 hour will be utilized for M phase. Okay, so 23 hours, somewhere about 95% is basically the cell is spending its own time only in interphase, and rest duration it will spend its time in M phase. Okay, that is another 5% it is going to spend time in M phase. Now the interphase is divided into three phases. G1 phase, it is also called as gap1 phase, S phase or synthesis phase, and G2 phase or gap2 phase. Now, individually, we look into what are these phases and understand much about the events which take place in G1, S, and G2 phase. So there are different names as I told you earlier. G1 phase is also called as gap1 phase. It's also called as first growth phase, or it is also called as post-mitotic, because it is happening after mitotic division, that is after M phase. So after M phase in interphase, the first phase will be G1 phase, then comes S phase, and then comes G2 phase, and one more M phase. So henceforth, it is after mitotic division, but it is before synthetic division or pre-synthetic phase. Gap between previous mitotic and beginning of DNA synthesis is the phase which is called as gap1 phase or growth1 phase. So what exactly are the events which take place in gap1 or growth1 phase? So the major events which take place is the synthesis of biomolecules. The biomolecules includes the carbohydrates, lipids, proteins, RNA are synthesized in this particular phase. Okay, so the major event which is taking place here is the synthesis of different biomolecules, that is carbohydrates, lipids, proteins, and RNA, which gets synthesized. So there is no change which occurs in DNA content of a cell. All right. So if the DNA content is the diploid, then the cell, even after G1 phase, at the end of G1 phase, it will be diploid in condition itself. Okay. So if it is haploid, if it is 1C, the content of the DNA, then it will be 1C itself. So it depends. Okay. So there is no change in the DNA content of a cell, but there is increase or synthesis in case of biomolecules. So that we need to focus on here. So since it is a synthesis phase, especially the biomolecules are getting synthesized, except DNA, you have to note it down, except DNA, because DNA is going to be synthesized in S phase. Except DNA, all other molecules are getting synthesized here. So synthesis of cellular components take place in this, except for the DNA. All other organelles, cell organelles are also produced, like mitochondria, Golgi apparatus, and then chloroplast, endoplasmic reticulum, lysosome. So all these are going to be synthesized. Cell organelles are going to be synthesized. Cell organelles are going to be synthesized, like mitochondria, endoplasmic reticulum, then comes Golgi apparatus, then lysosomes, chloroplast, if it is in case of plant cell, okay, etc. All these are going to be synthesized in this phase. Next one. Coming to the synthesis phase. So synthesis phase is also called as S phase. Okay. So S phase means what? S is nothing but synthesis phase. So what is going to be synthesized? We know that in the previous phase, that is G1 phase, except for DNA, all other cell organelles and the biomolecules got synthesized. It got accumulated, but except for DNA. So the DNA molecule will be synthesized in this phase. So hence it is called as synthesis phase. Okay. So DNA synthesis phase, we can even refer it to as DNA synthesis phase. And when we talk about the synthesis of DNA, each chromosome generally which will have two identical sister chromatids. So if it is a diploid cell, then it becomes tetraploid. If it is haploid, then it becomes diploid. So now if it is 1C, due to the duplication, it becomes 2C. If it is 2C, then the content of the DNA will become 4C, and it goes on. So since there is doubling in this process, there is doubling at the end of synthesis phase. So this phase is also called as replication phase because the DNA is going to double in its content. Okay, the DNA is going to double in its content. And also other than that, other than DNA, there will be synthesis of proteins which are associated with the DNA, which is called as histone proteins, which are necessary for the formation of protein in the cell. Okay, and there are few mRNAs which are also going to be synthesized, and nucleosomes are going to be synthesized in this phase. So nucleosome means a DNA which is covered by, a protein which is covered by the DNA is called as a nucleosome. Okay, it's called as a nucleosome. So we can say that the DNA along with the histone protein forms a nucleosome. DNA plus histone protein DNA plus histone protein gives us nucleosome. So the DNA is tightly packed in the histone protein and constitute the nucleosome. So the synthesis of mRNAs, the synthesis of DNA, the synthesis of histone protein and the nucleosome will take place in the synthesis phase, that is nothing but S phase. So coming to the generation of time, generally in case of the synthesis phase, it is about 6 to 8 hours or 8 to 9 hours. But in case of G1 phase, so let me go back and you can take it down. In case of G1 phase, out of 24 hours duration, 10 hours is going to be in G1 phase. So henceforth, this is the longest phase of interphase. Interphase on the whole is the longest phase in cell cycle, but in case of the interphase, G1 phase is the longest phase. And then comes the next phase, that is the synthesis phase, which lasts about 6 to 8 or 9 hours on an average. Okay, 8 or 9 hours on an average. Right. Then comes, yes, you can also see the duplication, how exactly it is happening. So these are the homologous chromosome. This is the centromere. So which holds the arms of the chromosomes, which are called as chromatids. So these are called as chromatids. These are the two chromatids. And each chromatid, when it undergoes duplication, it will be dividing. And that is how it forms a double. It forms a homologous chromosome. Okay, it forms a homologous chromosome. And this process of duplication or doubling is called as replication. Okay, the same replica will be formed. The same replica will be formed after the process of doubling. Okay, even here and even here. So that is how you can see the chromosomes which are going to be homologous. So if the content of the DNA here is 1C, then here it becomes 2C. If it is 1C here, it becomes 2C. If it is 2C here, then it becomes 4C and 4C, the content of the DNA. The chromosome number remains same. Okay, so the number of chromosomes, if it is 46 here, Even after replication, it will be 46. The chromosome number is same, but the content of the DNA, since the replication has undergone, it is going to be double. Okay. So what we need to note here is the chromosome number same. Chromosome number is same. It remains same, but the content of the DNA, is going to double. All right. So let's go ahead. Coming to the G2 phase. The G2 phase is also called as gap 2 phase, or it is also called as growth 2 phase. So this is a phase which is there after, which happens after gap, that is the synthesis phase, or before the YUM phase. So gap between DNA synthesis and nuclear division. Nuclear division is nothing but the YUM phase. So this phase is called as gap 2 or growth 2 phase. What exactly happens during this phase? So in this phase, RNA transcription and protein synthesis will continue. So generally, the DNA is going to, if this is a double-stranded DNA, the DNA is going to make its copies. Okay, it is going to make copies, and then it is going to form into mRNA. Later, the process of formation of DNA from DNA is called as replication. That is, it is going to replicate itself. The strands are going to replicate. And from DNA to mRNA, its synthesis is called as transcription. And mRNA is going to make some protein molecules. And that process is called as translation. So transcription, translation, and replication, these are together called as central dogma of molecular biology, which we'll be learning in the second year topic. But for you to understand, DNA is going to form its own copies called as replication. And from DNA, the message is going to be transcribed into mRNA, and mRNA will be forming into proteins. And this process is called as replication, transcription, and translation. Together, ultimately to form the proteins which are going to be used for cellular activities. It is going to be happening in both prokaryotic as well as in eukaryotic cells. So this process, replication, has already occurred in synthesis phase. So the next two phases will be the transcription phase and the translation phase. That is nothing but synthesis of mRNA and synthesis of protein will take place, or it will continue in G2 phase. Along with that, the other components of the cell organelles, the content of the cell organelles are going to be doubled. Okay, so there will be more intense cellular synthesis which will happen in case of G2 phase. So like proteins are going to be prepared more in number, which because the cell has to undergo the next phase, that is YUM phase. So where the cell will be undergoing prophase, metaphase, anaphase, and telophase, which is necessary for ultimately performing the growth. Okay, so henceforth, all the contents of the cell, the cellular organelles, goes on doubling. And proteins are also formed. Basically, the proteins which help in synthesis of spindle fibers are going to be born here, and asters are going to be synthesized. So this asters are going to be synthesized only in case of animal cell. Okay, only in case of animal cells. Likewise, even centrioles are going to be formed only in animal cells, in case of animal cells. And as we have to understand, G2 phase is the shortest phase in interphase. Okay, G2 phase is the shortest phase in interphase, which takes place about 2 to 5 hours. So overall, 23 out of 24 hours, 23 hours is going to be spent in case of interphase, and the rest 1 hour is going to be spent in YUM phase, or it is also called as mitotic phase. So this is the overall sketch of cell cycle. So you can see here the G1 phase. So G1 phase on the whole will last for about 10 to about 10 hours. S phase or synthesis phase, where the DNA is going to be doubled, and this will last for about 8 to 9 hours. And then the cells are now undergoing the preparation time where the doubling take place. So there will be more accumulation of the contents of the organelles for the cell division or cell multiplication. So henceforth, G2 phase will last for about 5 to 6 hours. Right? And apart from this, next phase which we'll be dealing with is the YUM phase. YUM phase is also called as mitotic phase in case of mitosis, or it is called as meiosis phase in case of meiosis. So it depends on the type of division which the cell is undergoing, right? So mitosis, here what exactly happens? In case of mitosis, we'll be understanding this in detail. In mitosis, the first phase after G2 phase is prophase, and then comes a transition phase which is called as pro-metaphase, and then comes the metaphase, and then the anaphase, and then telophases. Okay, so if you remember PMAT, then it would be useful for you to remember mitosis. So prophase, metaphase, anaphase, and telophase gives you the division of nucleus. So here in this process, mitotic division, the nucleus is going to divide. So when the nucleus divides, it is going to divide itself in 4 important phases that is called as prophase, metaphase, anaphase, and telophase. So the division of nucleus here, it is called as karyokinesis. Karyokinesis. The karyokinesis process will be followed by the division of cytoplasm, which is called as cytokinesis. Cyto means cell, kinesis means it is to divide. Okay, so karyokinesis is division of nucleus, cytokinesis is division of the cytoplasm. So the karyokinesis will always be followed by cytokinesis to complete one mitotic phase. Apart from all these, and you can see how the daughter cells are formed. So you can see the direction of the formation of daughter cells when a cell is undergoing the process of division. Apart from all these, there is one more phase which is called as G0 phase. Okay, now where exactly is this G0 phase? We'll understand what exactly is G0 phase. G0 phase is also, in other words, called as quiescent stage. It is also called as quiescent stage, Q-U-I-E-S-C-E-N-T, quiescent stage or quiescent phase. So this is a phase where the cell will not divide further. Okay, so what happens then if the cell is not undergoing under any division? Then this stage will be the cell will be entering this phase which is called as G0 phase. So in case of G0 phase, when exactly the cell will not undergo any division? When there is a injury which is happening to the cell. The conditions are like this: when a cell is undergoing any injury, okay, when a cell is undergoing any injury, or when a cell is undergone death, it has to be replaced, right? These cells which have undergone death, it has to be replaced. So it needs time for a cell to replace an injured cell or replace a dead cell. So henceforth, this phase where occurs is called as G0 phase, or is called as quiescent stage. For example, let's take the nerve cells or the skeletal muscle, the heart cells, maybe RBCs, WBCs, all these cells which have to undergo repair, or if it has undergone death, then it will enter the phase which is called as G0 phase or suspended stage, or it is also called as quiescent stage. In this quiescent stage, the cell will be metabolically active. In G0 phase, cells are metabolically active. That means what? The chemical reaction will be continuously happening, but there is no growth. Okay, metabolically active but no growth or no division. Okay, no growth or no division will occur. So generally, G0 phase is a phase which occurs. G0 phase is a phase which will occur from the cell cycle in early G1 phase. Okay, in early G1 phase, after cytokinesis, the cell may enter into a suspended stage which is called as G0 phase. So generally, in case of animal cells, on the whole, mitotic division will occur in somatic cells, that is the body cells. Somatic cells, that is the body cells. And in case of plants, the mitotic division will take place in both haploid and diploid cells. Okay, this is the G0 phase. So G0 phase is a phase which is called as quiescent stage, suspended stage, which take place during an injury or cell death. To replace it, it needs time. So until then, the G0 phase will be an important step. And apart from this, we need to understand that there are three important checkpoints in case of the whole cell. Okay, so there is and one more thing, this is also called as inactive stage. Inactive in the sense, only there is no growth or cell division, but there will be metabolically active, inactive stage. Right. Next one. So this is what I was telling you. There are three important checkpoints with regard to the cell cycle. We need to understand what are these checkpoints now. Okay. So these checkpoints are controlled or regulated by substances which are called as protein kinases. So I repeat, protein kinases means they are the substances which are controlled or regulated, okay, during cell division. So these proteins, the proteins in specific, they are called as cyclins. They are called as cyclins, or they are also called as CDKs or CDKs. What is CDKs? It is nothing but cyclin dependent protein kinases. Cyclin dependent protein kinases, they are called as CDKs, or they are also in short called as cyclins. So there are different types of cyclins for different stages of cell cycle. So each of them is synthesized at the required stage. Okay, and they are also destroyed at specific stages. So they can be created and they can also be destroyed. So when we talk about these checkpoints, what is the importance of these checkpoints? Because you know that the cellular activity which is continuous, which is simultaneous, but there should be something which has to check on whether the process is going on or not properly. Like in case of schools and institution, there will be teachers, and in turn there will be principal or headmaster or headmistress to check whether the process of whatever it is, the classes may be, or assembly, or anything which that sort will be properly regulating. Likewise, even in a cell which has to undergo different cellular processes, so there will be certain proteins which are going to control the cellular activity, and these are called as checkpoints. So there'll be checkpoints, and at this point the cyclins are created, and once it is checked, it is going to be destroyed. Okay, so there are three important checkpoints. One is found at G1 phase, okay, and the other one is found at the G2 phase, and the other one is found at M phase. Okay, so G1 phase checkpoint is generally if G1 phase, whether the organelles have been synthesized, whether the cellular components have been synthesized, will be checked by the G1 phase. Okay, G1 phase checkpoint will be checking whether the activities which are fine for the cell organelles which have already has been synthesized, except for DNA, whether the whole component have been synthesized is going to be checked by the checkpoint which is there at the G1 phase. Otherwise, if it is not, then it has an option of diverting itself and can enter into G0 phase. So it can enter into G0 phase. That means that it requires some more time for it to undergo the repair or any division. All right. So then comes the next checkpoint, that is at the end of this G2 phase. Okay. So in case of G2 phase, we need to understand whether the after S phase, whether in S phase the DNA replication has occurred or not. The DNA replication has occurred or not. So this will be further checked. Okay. So whether the DNA replication is proper or whether it has completed, that will be checked in the second checkpoint, that is after S phase in the G2 phase. Now the third checkpoint occurs at M phase, at the end of metaphase. Okay, this will be at the end of metaphase. Why it is going to check at the end of metaphase is because whether the chromosomes have attached to the spindle fibers or not, whether the chromosomes are aligned in the equatorial plane, so that when they divide, the components in the cell are going to be distributed equally to the daughter cells. That is why they are going to check at the end of M phase. Okay, and if everything is fine, then once again the cell cycle will be started, initiated from G1, then to G2, then S phase, and then G2 phase and M phase. The cell goes on undergoing the continuous cell division, ultimately for the growth and reproduction. Okay, so these are the three important checkpoints which have been regulated by the cyclins or CDKs. They are released and they can be even destroyed once they are completing their task. Lastly, so we spoke about M phase. We'll be dealing with the M phase in the next session, where we'll be understanding in detail about the M phase, the different phases which are included in mitotic division. So the last phase is cytokinesis. In case of cytokinesis, so cytokinesis is a phase where after the nuclear division, that is the karyokinesis, the formation of two daughter nuclei has been formed now. So this is followed by cytokinesis, where there will be equal distribution of organelles. This is what I told you. Even during the metaphase, the chromosomes are going to align themselves on the equatorial plane. Okay, so the chromosomes are going to be aligning themselves on the equatorial plane. So when they are going to be aligning themselves on the equatorial plane, just a second. So if this is a chromosome, they're going to be aligning themselves on the equatorial plane. So now when a cell is undergoing the division, the chromosomes are going to be separated. The chromatids are going to be separated. Along with that, even the organelles are going to be separated. Separated in the sense they are going to be doubled because they have already undergone in G2 phase. So now after the cell division, in the cytokinesis, there will be division of, if the cell has been together like this in telophase, now there will be division of the cytoplasm. There will be division of the cytoplasm. So this forms a new daughter cell, and this is going to form a new daughter cells. So that division of the cytoplasm is called as cytokinesis. Okay, division of the cytoplasm is called as cytokinesis. So cytokinesis plays a very important role because the cell wall is going to be separated. So the cell wall in case of plant cell is going to be separated in a different way, and in animal cell it is going to be separated in a different way. So henceforth the cytokinesis process is common for both plant cell as well as animal cell, but the way it is going to separate is going to be different in case of plant and animal cell. So in case of animal cell, it forms a cleavage in between, whereas in case of plant cell, it is going to form a cell wall plate or cell plate formation. So in case of animal cell, it is going to form cleavage. And in case of plant cell, it is undergoing and forming the two daughter cells by the formation of cell plate in between the two daughter cells. So that is the difference between the separation technique which occurs in case of cytokinesis. All right. So in the microscopic view, you can make out this is the interface. So here the cells are in each cell. If this is a plant cell, so this is a plant cell which has the cell wall, and you can make out here in the nucleus. In the nucleus, there is a dark portion which is called as nucleolus, and then inside this, now the interface is occurring, okay, which includes the G1 phase, S phase, and G2 phase, followed by the M phase, okay, followed by M phase. Now, overall, we have to understand, why do cells divide? For various purposes. We know that, right? We know the reason that a cell divides for growth. The cell has to divide for development. The cell has to divide for repair of wound tissues and reproduction. And also, we need to understand the importance of cell division. That is, it facilitates the exchange of material, maybe the genetic exchange or physiological exchange, the cellular exchange. So at every level, it will help in facilitating the exchange of materials. And overall, cell has to undergo the division. Otherwise, you know that if the cell is undergoing the division here itself, and then the DNA is also going to be synthesized and the organelles are going to be replicating. I mean, the DNA is going to be replicating, the organelles are going to be synthesizing more and more. The cell will be obviously rupturing. So there will be no continuation of progeny as such. So to control all these processes, including the controlling of DNA overloading, the cell has to undergo the process of cell division. All right. So coming to the phases of mitosis, we'll just understand what exactly is mitosis. Mitosis, mitos means thread, osis means state. So here in the mitosis division, it is also called as equational division. Mitosis is also called as equational division. So even we know that interphase is divided into three substages like G1 phase, S phase, and G2 phase. Like that, even mitosis is divided into four important phases, that is PMAT, as I told you, prophase, metaphase, anaphase, and telophase, which are the important stages of mitotic division. Okay, why it is called as equational division and what exactly are the events which take place in the mitosis is what we have to learn in the coming sessions. Now let us solve some of the questions based on the topic which we have discussed, that is the cell cycle. The first question, the stage at which DNA or chromosome replication occurs. We know that telophase, prophase, metaphase are the stages of mitotic division. DNA or chromosome replication will occur in S phase, which is also called as synthesis phase, So synthesis phase is a part of interphase. So henceforth, our option will be option B. Okay, it will be option B. Next one, the sequence of cell cycle is. So we need to know the sequence of cycle, whether it starts from M, G1, G2, S phase. The option B is G1, G2, S and M phase. Or the option is S, M, G1 and G2 phase. And lastly, G1 phase, S phase, G2 phase, and M phase. So we know that the sequential events which is going to take place is starting from G1 phase, that is gap 1 phase, synthesis phase, growth 2 or gap 2 phase, and later M phase. So henceforth, the option will be option D. Next question. The stage of cell cycle when cell has undergone differentiation. So when the cell will undergo the process of differentiation, because during certain conditions like cell repair, cell loss, so in order to incur the damage which is caused due to the cell at various stages of the cell cycle, so it will undergo a process which is called as G0 phase, right? G0 phase is also called as quiescent stage, quiescent stage, or it is also called as inactive stage, inactive stage or inactive phase, right? G1 phase, there is no G3 and G4 phases. There is only G1, G2 and S phase. So these three options, these two options are wrong. And even this option is not correct because it will not undergo any process of differentiation. So henceforth, our option will be G0 phase. Next, the longest phase of cell cycle is, see, the question may be asked like this. Either they'll ask you the longest phase of cell cycle, the longest phase of interphase, or sometimes they may ask the longest phase of M phase, or the longest phase of meiosis 1 phase, or on the whole the cell cycle. So it is different for different phases. So on the whole, here they have asked the longest phase of cell cycle. So we know that the cell cycle is divided into interphase and M phase. So interphase is said to be the longest because it takes place about 23 hours. 95% is because of the interphase, and rest 5%, around 5%, which is contributing for 1 hour, is nothing but the M phase. So since the options here is prophase, of course, in case of M phase, the prophase is the longest phase. In case of interphase, G1 phase is the longest phase. Okay. So here the option here is the longest phase, which is in case of cell cycle. So henceforth, our option should be option longest phase of cell cycle. They have not mentioned which cell cycle. Of course, since they have not mentioned, it has to be G1 phase. Okay. So even though interphase is the longest phase among M phase and interphase, so the G1 phase, we have to choose the answer. Next one. Cells in G0 phase of cell cycle. What happens to the cells in G0 phase of the cell cycle? They exit cell cycle, they enter cell cycle, they terminate cell cycle, they suspend cell cycle. So what happens, you know that after G1 phase, so from the G1 phase, there takes a deviation and enters a quiescent stage, which is called as G0 phase. So during this phase, they are metabolically active, but there is no cell growth or division which is taking place. So they don't, they exit the cell cycle, they enter the cell cycle, they terminate the cell cycle, they suspend the cell cycle. So they are going to exit the cell cycle, but whenever there is an opportunity, once again, when everything is being the repair, everything has undergone, the process has undergone, then once again they enter into the cell cycle. So here the option is they exit but they return. They do not enter, they terminate, they suspend. They are going to suspend the cell cycle for a meanwhile, but they are going to come back. Okay, so it cannot be exited completely. So they are going to be suspending their activity for a meanwhile or time being, and then they are going to come back. Henceforth, the option will be option D. The term karyokinesis is used for. So the term karyokinesis is used for disappearance of nuclear membrane during metaphase, changes occurring at anaphase when chromosomes move to opposite poles, event occurring during interphase, overall changes occurring in the nucleus during cell division. So disappearance of the nuclear membrane during metaphase, since The metaphase is belonging to M phase, so it is a different stage. Changes occurring at anaphase when chromosomes move to opposite poles. This is also one of the phase which occurs in M phase. Events occurring during interphase. We know that interphase is divided into G1 phase, S phase, and G2 phase. So henceforth, even this cannot be option. The option here is overall changes occurring in the nucleus during the cell cycle. We know that the karyokinesis is the changes which is happening, okay, which is further divided into PMAT, and one more stage is cytokinesis. So nucleus is undergoing a lot of changes here during the cell division. Henceforth, the option here will be option D. Okay, it's option D. Next question. In the cell cycle during interphase, what exactly happens? So during interphase, two daughter cells are produced. During interphase, we know that after end of mitosis, the two daughter cells are going to be produced. Okay, so two daughter cells are produced. The nucleus is divided into two daughter nuclei. The chromosome condenses, the DNA is replicated. Two daughter cells are produced at end of mitosis. So this statement is wrong. The nucleus is divided into two daughter cells. That is what this nucleus is going to divide into two daughter cells. So during karyokinesis of mitosis stage, the chromosome condenses. Even the condensation of chromosome will take place in the M phase, whereas DNA is replicated. So the replication takes place here. That is nothing but the synthesis phase, S phase. So henceforth, option should be option D. Next question. The amount of DNA per cell is dash based on the initial amount. So what happens to the DNA per cell? So the amount of the DNA, if it is 1C in case of the parental cell, what happens to the cell later? If it is 2C, what happens to the cell later? We all know that the amount of DNA per cell at G1 phase, if it is 1C, 2C, at synthesis phase, at the end of synthesis phase, it is going to get doubled. It is going to get doubled because there will be a single chromosome which is going to double itself. So henceforth, 1C becomes 2C and 2C becomes 4C. Henceforth, the option here is it doubles. It will not triple. There is no change. Obviously, change will occur. It decreases. No, it will increase. So henceforth, the option here is option A. Next question. The number of chromosomes in onion root tip cell at G1 phase will be what? So first we need to understand what will be the number of chromosome in onion root tip cell, especially in a plant cell. Even in plant cell, the number of chromosome goes on varying. Okay, the number of chromosome goes on varying. So in case of, let's look into the chromosomal number. So the chromosomal number for an onion root tip will be 16. Okay, it's a diploid number. So at G1, we know that the chromosomal number will be the same, isn't it? The chromosomal number will be 16. Even in G2 phase, even in S phase, the chromosomal number will be 16. Here we are talking about the number of chromosomes. We are not talking about the content of the DNA. Okay, so the number of chromosomes at S phase will be 16 itself. Whereas in case of G2 phase, even though there is no doubling of the chromosome number, it will be 16 itself, right? But when we talk about the that is the DNA content, Generally, if it is at G1, it is 2C. At S phase, it becomes 4C. And at the end of G2 phase, it will be 4C itself. Okay. So this is regarding the DNA content, and this will be regarding the chromosomal number. Here, the number of chromosomes in onion root tip cell at G1 phase will be 16 itself. Okay, next one. In interphase in human cell, lasts for, we know that interphase is a phase which it is the longest phase, and it takes place about 23 hours. Here the option is 22 hours, 95% of the cell, 24 hours, around 90% of the duration of the cell. It is 23 hours, that is somewhere it is constituting about 95% of the cell cycle. Henceforth, it is option B. So coming to the next question, incorrect statement with respect to cell cycle. So which is the incorrect statement? Duplication of genes occurs twice in meiosis. No, there is no duplication of genes, because duplication is nothing but in case of synthesis phase. So synthesis phase will take place only once. Okay, there is only once there will be duplication. So henceforth, the first option is the incorrect statement. Okay, so that means 1C cannot be becoming into 2C, and 2C cannot become into 4C in only S phase. So here the statement is duplication of genes occurs twice. This is one time, this is one time. So it cannot be possible. So henceforth, it is only either 1C becomes 2C or 2C becomes 4C. The rest other statements are true, because karyokinesis occurs twice during meiotic division. Yes. Cyclins are proteins that activate protein kinesis to regulate the cell cycle. Yes. After telophase 1, chromosome number is reduced to half because it is undergoing meiotic division. Right. So we have completed today's session with the cell cycle. In the next session, we'll look into more topics. Thank you, students.